Good morning everybody. Today I am giving technical seminar on protein based storage systems. Protein based storage system, this is a theory proposal was processed by Dr. Robert Brigade in 1992. It is a molecular electronical is broadly defined as an encoding and manipulations and retrieval of the informations in the molecular or macromolecular level. This is approach contrasts with the current techniques. It's a material to generate integrated circuits. A advantage of molecular approach is the ability to design fabricate device in bottom up and an atom to atom theory. A molecular computer cloud in principle be 2 to 3 orders magnitude and smaller and faster presence of semiconductor conductor composed of a compatible number of logic gates. However, extrapolations of Moore's law predict that the semiconductor future sizes will be the molecular domain around 2030. There are the two possible scenarios. The first, Moore's law remain valid until 2030 and lithological technology will evolve to create nanoscale device with the much scale of those currently involves the molecular technology. The second scenario, the semiconductor evolutions will hit a wall, the economic reasons. Memories by nature in computers have become the assessors. Earlier technologies utilized the processing speed for problem solving. The first computer had both memory as space and constants. Problem solving on the computer requirements both need to improve the time and space of a complexity. A complex jargon at a time it takes to solve a problem based on the parameters of input solving problems algorithm, while development in the space of allowing being the overcome the time complexity remain issues. However, in the terms of long term and the memory, a bandwidth latency considerably improvements are the seen. Various improvements are to be seen, but the molecular electronic support of the bacteria rheoordopsin seems highly promising. In the theory, practically movement of 1000-fold is expected. Practical demonstrations have their improvement its improvement about 300. The key aspects of rhodopsin its ability to be photonic device of a photon pump. It's quickly these transmissions take place based on the quality of incident light. This is properties of the systems provide for a larger number of states. It will help for working the large storage. Protein memory is experimentally is a means of storing data. Using the proteins, the response to light from bacteria found in the salt water, the small cube can be stored the large amount. It's the main that what the robot brigades is tell us means it's a very large data can be used by the protein memory systems, which is a biodegradable, like we can use the huge amount of data within the very small amount of water. By the using the laser protein can be changed depending on the various waves length, allowing them to store the recall the data. As a result, the protein can be stored in enormous amounts of using the laser to write and record binary code. They discovered as the protein isolated from the marsh, the bacterium exhibited and photosensitive property. They called this a protein bacteria rheoordopsin because it is very similar to the protein ordopsin that finds in the eyes of humans, eyes and animals. The bacteria rheoordopsin it is also called as a protein memory systems. Data writing techniques is a photocycle. The slide will explain how the data writing techniques is carried out in the protein memory systems. The bacteriorhodopsin has the 7 transmission membrane or helicas that make up the proteins as a secondary structure. The productions of a purple membrane allows the organisms to switch the photosynthesis as a means of energy productions. When they dissolve, the oxygen concentrations drop below level of sufficiency to sustain respective oxidative phosphorylation. The light absorptions of chromophore, the light adopted from the protein La or simple is a transmitter oriental polyphenol carbon atoms or a cooled orange. Covalently, the bonds of a protoponderate skiffon base linkage. The absorptions of light initiates the complex of a photochemical cycle approximately 10 milliseconds in the duration. As here, the laser is a transmitter into the memory cube and the detector, the green laser sense is a laser array. These are the mainly divide the memory systems which allow the storage in the memory cube. The photocycle transposes a proton from the
internuclear extranuclear sites of the membrane. The light introduces the proton pumping that generates the electrochemical, and the gradient that the bacterium uses chemostically synthesize ATP. Photochemical stability is quantified as number of times the photochromic material can be photoconverted between the two spaces before the 37% of the irradiation assembled in edges. This number is the 37% of irradiations assembled in edges. This is a number is called as a cyclicity. It is extends 106 for bacteriospins at ambient temperature. Few organic photochromic material approaches this value. Those that have high value cyclicity typically have low quantum of efficiency of photoconversions and photoconversions. The data reading techniques of a photocycle, which is here, there are the same thing as in writing lasers. Light lasers is absorption, and green lasers will also be a retrieval of data from the BR code. The laser arrays, the memory can be absorbed, and the detector is always detecting the light sensors. Select the absorptions of the light array from the zero internet data states, and which has to be make the photocycles as same in the writing cycles photocycles. Here we know the high cyclist of a BR is due to chromophorming from the reactive of oxygen and singlet oxygen and the free radicals. The common devices is not a bacteriospin. The absorptions of a spectra of the keep photocycle intermediates and the branching proteins. And here the absorptions of the spectrum of each intermediate due to changes of geometrical chromophones as well as the changes of binding sites. And detail the molecular origins remains to be determined. Even the resting state, the electrostatic natures of a binding site has to be remain the subjects of controversy. A majority of evidence suggests that the counter ion, the forming of a two positive charges separate by two negative residues, aspa 85 and aspa 215, through the genetic engineering and the chemical modifications are dispressive for electrostatic and propositive, and site can be yield analogies and the proteins with a different spectroscopia. This is the execution process of a photochemical event. The primarily photochemical event light adapted bacteriospins involved in the two subsequent process where that is the first produce executions of the chromophore to produce the photovoltaic conservations due to loss shift of electron density accommodations, and the shift in the electron density is a moving of electron around the 2.5 amps the down to the polyphene chain towards the nitrogen atom, and this shifts and is a linkage of memory negative. Once the favorable electronic reactions is between the C14 and C15H, it's a neon will be added, and the two nearby negative aspartic acid is produces, while the bacteria and the photons will be attached, and we are producing electrostatic reactions, and we got the negative of aspartic acid. Now the repulsive and these interactions helps direct in the photoisomerization and 13 CSC double bond to generate the primary photochemistry. The photostatic interactions is a coupled of a significant bond order the reversal due to excretions of a process and sufficiently generate nearer by the barrierless executed. And the potential that dramatically the existence in the state and characterize biological equivalent high electron and mobility transistors. The photoisomerism electrostatically selected because the protonations of aspa 85 changes and excited the surface of dramatically, and makes the formations of 9 CSC photoproduct competitive with a 13 CSI photoproduct. This is a possibility exploited the branched photocycle volumetrical memory. And here this is a process. Data writing and reading is in the parallel process. The main advantage of the bacteriospins is mainly it's write in parallelly. As we know that parallel applications is also faster in the reading and as well as in writing. And in the both selections activations of thin segments of protein within the volumetric memory medium, a thin activated region is called as a page, and the activation process is also classed as paging. The thickness of the page is dependent on the optical arrangements of various forms of RMS value of 15 micrometers, multiple of present systems, and 200 micrometers. The simply cylindrical lens. Data are written in the using the orthogonal beam. The light is shifted in the times is to photo activities. The O state, the process explicitly documented on the left hand side, as we shows in the figure, and the process that written 3 bytes within the page. The vertical axis of the nominal time relative to the firing the page addressing is a paging laser. 2 milliseconds of following paging, the data laser and the data beam spatially light modulated. To irradiation those volumes elements into which 1 bits written in that. The process converts of O to P, these so only that these O excel the memory covert. The volume eliminated and created single write pixels only with. After the many times P state suddenly decays up to the Q state, and the write process accomplished and the time takes in the right beam as shown, and the beam is taking the simultaneously but the simultaneous convenience, the use of prisms and narrow page and the near diffractions limited but the expense of complexity. Only one side paging optics show in the description process writing in the single bit as well as the methods of available storage density. The data are read parallelly and different absorption. In the photobacteria reordospins, in this memory data can be erased by the page globally. Here the blue lights of the total one axis coherent light individual pages can be cleared by the using the irradiations at or near the 410 nm wavelength. The intercepts of both P and Q, our earlier prototypes using the 413 nm output and kyratospin laser. Such approach this is usefully for the level of one prototype. However, the various of second prototype investigated the 3 alternatives. They can clear the entire data curve using the incoherent light of 316 nm and 450 range. We explore this is the process of prototype describes the below. The broadband and clear the operations must be carried out. The absence of red light, however, because of a blue light activated by photocycles produces the O state. As we one reviewer the noted the O state is approximately and the 362 340 and the range. This is a situation does not create a causes problem. However, because the absence of red light limits the populations of a O state molecule is a small amount generated by the photochemical conversions of a P because of the and P and Q states or preferentially absorbs the erase beam. The end of a photosynthesis, the state is the distortions of a favor of Beer factor, and how many hundreds of arrays are efficient. Eraser does not have the perfect, however, the memory works properly. And the concluding here, the remarks is the whether the devices describes here commercially the futures and applications of a bacteria reordospins, the mainly ultra fast RAMs. The RAMs is a very faster memory which can be used for the main memory of systems. The ultra fast RAMs for the using of the faster executions of a data. And another applications of a bacteria reordospin is a fingerprint processing. As we know, the fingerprint processing is by the taking of the thumb impressions, and as well as it's a very time complexity applications which is by the using of bacteria reordospins memory we can do this process with a faster manner. And the fingerprinted processing memory is by taking the thumb impressions as well as and we can do the DNA computers DNA applications also by the using of a bacteria reordospins is a very faster manner. Here we are using the optical switches and the neurological gates. As we know that neurological gates, it's a very complexity of neurons, and we have to take the total of genetic engineering as well. Very taking the one DNA and as well as we have 4 DNAs. We can make them separate. It needs a huge amount of memory. Then here the bacteria reordospins memory will help you very much important. Bacteria reordospin is an advanced manner of memory systems. Here the present status of bacteria reordospins, it's not used for the commercial applications. It is only now under the development.
and it is mainly used for the military and scientific applications. Research is going on for this bacteriospins, high speed of high capacity memorial for the commercial applications. It is not still now it's not in the commercial, as we know that, and it's a very huge amount of memory it will provided for you, and the ultimate mission intelligence with the acid of genetic engineering, the memory of mnemonics of human brain. As we know, the neurological means the neuro memory system is a very huge memory system. By the solving these problems, we need so much large memory of memory, so that the bacteriospins memory, which will help it for you the convert the total neuron memory as a very in a simple manner, the carrying the small encyclopedic cube containing all the informations we need. The conclusion, we know that during the past decades of the speed of computer process increased almost 1000 times, where the data storage capacity is increased only by the factor of 50. Also, the transfer of the data within the computer remains the principle of a bottleneck, the limits of a performance. And the bacterial devices that generated sufficient inertia to the cause of unjustified preoccupations with a protein. We believe that both statements are correct. Bacteriospins is unique in the providing of thermally stable, photochemically reused, easily modified proteins that can be used for the active component in a variant photonic device. The evolutionary development, the light translating the protein is a slash match of a bacterium, selects the number of properties of that's required for the devices of applications of that investigations here. But it is also true that for too little effort has been directed at investigations of other proteins and these the alternatives of biomaterial, and may ultimately provide the superior performance. We hope that article will be help us and simulate not only consume in the bacteriospin, but renews the interest of exploring the proteins for the device applications. The protein memories uses the laser beams, which improves their life with the reductions of wear and tear. As like bacteriospins renew the interest exploring and the proteins device the applications. The main our conclusion, the bacteriospins very huge amount of applications of memory it can be supported, and the high performance can be also used, and the main bacteriospin is not used for the secondary memory, it's ultra fast RAM.